Hi, in this video I will demonstrate how to calculate and analyze biodiversity using QIIME 2. Our experiment's data contains 23 item samples plus 3 negative controls. The 23 samples are personal items of 5 different people from their inside of shoes, wallet, phone screen, water bottle mouth, and glasses. This is the workflow. So you can see that the 3 key stages. The first stage is pre-processing the data, and then the second stage is building the phylogenetic tree and also taxonomy assignments. And then the last section will do the biodiversity metric calculation and also analyze the data. So to begin with, let's look at our input data. We need 2 types of input data, being the sequence file and the metadata spreadsheet. We'll look at the sequence file first. So again, the sequence file contains 23 items from 5 people plus 3 negative controls. The samples were collected and then it's put through in the wet lab to do the DNA extraction. So the 16S amplicon sequencing using Illumina Me-SEQ is used to extract DNA. Now what that 16S amplicon sequencing means is that the 16S rRNA gene sequence is used to classify all prokaryotes. And the 16S rRNA amplicon sequencing is an application of the 16S rRNA sequence-based classification to characterize biodiversity and to investigate the ecological characteristics of all sample types. The benefit of the 16S rRNA amplicon sequencing is that comparison can be made to the extremely detailed and well-curated taxonomic databases. Now let's look at our metadata. This is our metadata. The attributes we have for the samples include the item type, person, age, and ethnicity, and sex. So obviously, sex, age, and ethnicity is to do with the person which the item belongs to, not the item itself. When the input data is ready, we can start our data pre-processing. So we'll be using QIIME 2 to process the data. We use QIIME 2 through the Jupyter Notebook in Bash kernel and Anaconda. So these are the codes to load Anaconda and activate QIIME. And in the workflow, you will produce many .qza and .qzv files. And the .qzv files are for visualization in the QIIME 2 website. So this is the QIIME. So that's the QIIME 2 website, and it looks like this. So for all the .qzv file that you produce in the workflow, you can drag and drop it in here, and it will automatically produce visualizations that are relevant to your file. So these are the codes we use to import our data. Note that our data is already demultiplexed. So from the summary of the data, so this is the codes we use for the summary of data, and it will produce a .qza file and a .qzv file. So once you've run this command, you want to drag and drop the .qzv file to the QIIME website, and you'll get a summary that looks like this. So note that we start with more than 6.5 million sequences in the data, and this is at a sample level summary. Now note that because some of the samples were not able to produce data, we only end up with 20 samples in our data, so our analysis only have 20 samples. Okay, now we want to look at the quality of the reads. We want to first examine the quality of the reads and exclude low quality reads in our analysis. So denoising is a quality filtering of individual reads to remove low quality reads, and it can also include the removal of chimeras, foreign DNA, and individual bases with low quality scores. So the chart on the right here shows the quality of the reads, which is in the y-axis, by sequence space, which is in the x-axis. You'll see that the confidence of the quality drops as the sequence gets longer. You can see that. Okay, so how to do denoising? The first part, the most important decision to make, is to select the sequence base where the cutoff where you're happy with the quality of the reads. So just a little bit more about the quality score here. This is the Phred score, and it reflects how confidently the right bases have been called. The Phred score of 10 corresponds to 1 error in every 10 base calls or 90% accuracy, and a Phred score of 20 corresponds to 99% accuracy. There is no defined cutoff on how low an acceptable Phred score can be, but one should aim for a Phred score higher than 33. So and then what you see that these lines here are actually individual box plots at each sequence base. So the darker line here, it's the box plot itself, and the lighter line here, it's the whisker of the box plot. So we want to select somewhere like a region that the Phred score it's between 20 and 30, I guess. So this is what we've done here. We're not aiming for just 33 because it will be very low, like very little, but you can see that most of the box plots here, it's more than 35 and there's some whiskers. So we can allow for a little bit of error because that will be the data will be further cleansed in the following steps. So this is the same for the reverse read. So the forward read will select a sequence space of 160. And we'll do the same for the reverse read. 160 also works quite well here. And so the filter chosen, it's 160 for the forward reads and the reverse reads. And we'll use data 2 to denoise the data. So these are the codes. And this is where we put in the chosen filter, these lines here. So the -F means that's for the forward read, and the -R means that's a cutoff for the reverse read. Note that this step can take over 7 hours to run, so just be careful before you click run. So after we have processed the data, these are some of the results you will see in your Jupyter Notebook output. So that's the sample. So let's look at sample AP11. So at input, so we have 337,000 sequences at input. After it's filtered, it's reduced to 280,000. After it's denoised, it remained at 280,000. After it's merged, it's reduced to 1,377 sequences. And after we've removed the chimera, it's reduced to 1,052 sequences. So that's quite a dramatic drop, but it's mostly to do with the merging. And you can see that for each of the samples, they've run that. So now that the file is cleansed, we want to have a look at the summary of the file. And you can see that, so that's the code you can run to get the summary. And we can see that now we have 13,000 sequences in the cleansed file from the 6.5 million. So looking at the sample AP11 again, so as you can see, it's reduced to 1,052 sequences. Now we want to move to taxonomy and building the phylogenetic tree. So it's important to build the phylogenetic tree first because both alpha and beta diversity, a lot of the metric uses the phylogenetic tree. So the steps we would take to create the phylogenetic tree include: number 1, we want to perform multiple alignment to further reduce the noise and prepare the data for phylogenetic analysis. So green genes is used as the standard database for alignment here. Then we'll perform alignment masking. So that eliminates phylogenetically uninformative or misleading sites from an alignment before phylogenetic analysis. This is common practice in phylogenetic analysis. And the third, we want to build the phylogenetic tree. So these are the codes we use to build the phylogenetic tree. So first part, it's multiple alignment, so we use math here. And then we want to do alignment masking, which we use mask here. And then we use fast tree to build the tree. Now note that this tree, when it's built, is unrooted. So what we need to do is to assign the roots for the tree. And in here, we have decided to assign it at the midpoint of the tree as the root. After we've built the phylogenetic tree, we need to assign taxonomy. So we'll use the feature classifier, classify sklearn, and this file is actually a pre-trained classifier for bacterial DNA annotated to the green genes database for taxonomic assignment. Okay, so we've built a tree, but we don't know what the taxons are, and the taxonomy assignment will tell us what the taxons are in the tree. So after we've run the code, we can visualize it and see that, well, these are the feature ID that we've identified in the data, and for each feature ID, we've now assigned a taxon. So for this file, we've only either have unassigned or K bacteria in the features as taxons. This is a bar plot for the taxon. So this is by item type and this one by person. The darker blue, it's for bacteria, and the lighter blue, it's unassigned. So what is quite scary is we can see here glasses actually has more bacteria than some of the phones, shoes, and wallet and water bottle mouth, which is quite scary. And then this one, it's the again the taxonomy bar plot by the taxon bar plot by person. Now that the data is ready, we have both the phylogenetic tree and have assigned taxon. We will now go into calculating the biodiversity. Again, one more step before we actually calculate the metric for the biodiversity is to consider the sequence depth for biodiversity. So different sequence depth will provide different results for our measures. What we first need to do is to explore if we have sequence depth enough to capture the diversity in the sample. And we'll do this by using the rarefaction curve. The rarefaction curve analysis is performed to see if some samples are represented with low sequencing depths. Based on the curves, we will have to identify a cutoff read depth for alpha diversity analysis. Note that samples below the particular threshold that we choose will not be included for further analysis. So generally, we want to choose the smallest sample size for the rarefaction curve. However, our samples are already very small. So you can see that in here, the sequence depth for the samples, the lowest is only 104, which is really low. So we'll need to try a few different cutoff for the rarefaction curve. Now, the thing is, and if we select a higher one, we'll have to start excluding some of the samples. So we need to choose, I guess, a point that sort of balances both capturing biodiversity and keeping as many samples in the data as possible. Okay, so these are the codes we use to run the rarefaction curves. And what this is calculated. It calculates the alpha diversity for faith, and then also you can choose that for Shannon. So you just choose it from this drop box here, and you can see that the curves. The Shannon is a good example where at 50 basis point you can see that it plateaus, so there's no more increase. So what you don't want to do is select somewhere that's along this line because the biodiversity is still increasing. But then it will be good to select somewhere it's sort of the lowest point where it starts to plateau, and that will give us good biodiversity information. Now it's kind of easier for Shannon because it doesn't require a lot of depth, but for Faith we can see that 100 is nowhere near enough. Neither is 300 because the curve is still increasing. At 500 it starts to get a little bit better, so you see a little bit sort of trend of plateau. It's not fully plateaued, but I guess it is what it is. And it's interesting to see this blue curve here because there's a sudden jump in biodiversity, so which is quite important to capture. So what we'll do is we'll select a cutoff at 500. These are the codes we use to calculate all the biodiversity core metric. So you just need to do this. Put in your sampling depth, which is the threshold we've just identified. We'll use 500 here. Once you've run this command, it will produce a list of, or a list of core metric that's already calculated for you. Some of them already comes with the visualization file. Some of them you have to run an extra step to convert them into visualizations. So let's look at the alpha diversity. So alpha diversity, it's a diversity within a particular area or ecosystem, usually expressed by the number of species in that ecosystem. We'll consider two alpha diversity measures in our analysis, being the Faith's phylogenetic diversity, where phylogenetic tree is incorporated, and the Shannon's diversity, which is an index where both richness and evenness are taken into account. So again, these are the files we got from the previous step in the core metric calculation, and these commands are just to convert them into a visualization file. So this is the result for the Faith diversity, the box plot, and the p-value. So note that there's the overall p-value, it's 0.07, so it's not significant, so we'll use the benchmark as 0.05. And in this case 0.07 is not significant, and in all the analysis bear in mind that our sample size is really small, so just keep that in mind. So when we look at the pairwise comparison, we can say that maybe between wallet and water bottle it is sort of just below the 0.05 benchmark, which is borderline. So there's probably more alpha diversity between the wallet and the water bottle. Don't worry about this. So some people may choose to relax it to have just less than 0.01 and accept it, but really in our case the sample is really small, so don't worry about that. So then this is the Shannon diversity index, and similar results. So there's the diversity is not significant. Then at most you can say between wallet and water bottle, then there is some alpha diversity between the water bottle and the wallet. And then we look at the beta diversity. Beta diversity is a comparison of diversity between ecosystems, usually measured as the amount of species change between ecosystems. We'll consider two beta diversity measures here. We have the weighted unifrac, which measures the presence and absence of taxa and incorporates the abundance of taxa. There is the Bray-Curtis, which calculates the dissimilarity in samples composition, but it's not based on phylogeny. So these are the codes. Again, that's from the previous step. What you need to enter is the the attribute here you want to get for the beta diversity. So in here, we want to look at for item type. Now, unfortunately, there's an error, and that is because some items only have one sample after we have set the sequence step cutoff to 500. So for demonstration purpose, we'll continue the analysis, but we'll look at the beta diversity between gender. This is the beta diversity for unweighted unifrac, and that's you can see there's only one dot for shoe, it's only one dot for phone, etc. So that's why we can't really do that. Doesn't come up with like I guess a p-value for it, but you can still visualize it. So this is the result if we look at the beta diversity by sex, and that's the p-value, which is really high, so there's no significance in the differences in samples by sex. And then you can see here that's a visualization of the unweighted, of the weighted unifrac, and they just cut it together. So you can see that's why we conclude that there's no really significant difference between sex. And similarly for Bray-Curtis, so that p-value is really high, so there's no significance in the difference in sample by sex and the visualization of that. So what you can do is for all the metric that's calculated from this step, you can do a visualization and repeat the analysis. So basically look at the p-value, and you can also look at the visualization on how different they are. So that concludes our demonstration for today, and thank you for your time.